The human eye does not passively record the world like a strip of film. It is a biological transducer, an organ specifically engineered to capture visible electromagnetic radiation and convert it into a neural code the brain can process. Our paired eyes sit protected within the orbits, the bony sockets of the skull. Inside rests the adult human eyeball, a nearly perfect spherical structure. Operating this structure presents a severe engineering challenge. The eye must maintain strict structural integrity to hold its shape. It requires constant vascular nourishment to keep its tissues alive, and yet, simultaneously, it must maintain total optical transparency across its central axis so light can pass through uninterrupted. The solution to this challenge is a tripartite structural foundation. The wall of the eye is built in three distinct layers: an external fibrous tunic for structure, a middle vascular tunic for nourishment, and an internal neural tunic for sensory processing. Vision, fundamentally, is a highly sequential process. It begins with the physical lensing of light through the outer layers and ends with a paradoxical inside-out molecular dissociation deep within the neural layer. By tracing the path of a single photon through this exact structural hierarchy, we can derive the functional anatomy of the entire organ. Isolating each layer reveals how biological tissue organizes itself to solve complex problems of physics and optics, achieving results that mimic high-precision engineering. Looking at this cross-sectional diagram of the eye, we start at the external layer. This is the sclera, composed of dense connective tissue. It forms the tough, white outer shell that protects the inner mechanics. As the sclera moves to the anterior portion of the eye, it transitions into the cornea. This is the primary refractive surface, the clear window where light first enters. To remain perfectly transparent, the cornea must meet a strict structural requirement. It contains absolutely no blood vessels. As noted in the handwritten margins of this page, the physiological consequence of being completely avascular is that the cornea must absorb oxygen and exchange nutrients directly with the outside environment. Moving inward, we find the middle layer, the choroid. In contrast to the cornea, the choroid contains a dense network of blood vessels that nourish the eye, and it appears distinctly bluish in color due to its pigmentation. The choroid is thin over the posterior two-thirds of the eyeball, but as it reaches the anterior section, it thickens significantly to form the ciliary body. This ciliary body continues forward to form a pigmented, opaque structure known as the iris. The iris is the visible colored portion of the eye you see in a mirror. Its primary mechanical function is to regulate the diameter of the pupil, controlling exactly how much light is allowed deeper into the system. Positioned immediately behind the pupil in the iris is the transparent crystalline lens. The lens doesn't float freely. It is suspended in place by ligaments that are directly attached to the surrounding ciliary body. The placement of the lens naturally divides the interior of the eye into two separate fluid-filled spaces: the anterior aqueous chamber in front and the posterior vitreous chamber behind it. The smaller anterior chamber contains aqueous humor, a thin, watery fluid. The much larger space behind the lens is filled with vitreous humor, a transparent, gel-like substance that maintains the shape of the globe. So the physical path of incoming light is direct. It passes through the cornea, crosses the aqueous chamber, travels through the crystalline lens, and crosses the vitreous humor. Every millimeter of this macro architecture is optimized for a single objective: minimizing the scattering of light and maximizing its sharp focus onto the posterior wall of the eyeball. It is only after completing this journey through the optical mechanics that the light finally strikes the deepest and most biologically complex layer, the retina. The external and middle layers serve as a preparatory stage. They do the heavy lifting of physical refraction, delivering a tightly focused beam of raw light directly into the neural matrix. The retina is the internal neural tunic. This is the layer that actually houses the photosensitive cells required to detect light. Looking at this retinal cross-section diagram reveals a paradox. The cellular hierarchy is arranged inside to outside. Tracing the handwritten notes, incoming light must first pass through ganglion cells, then bipolar cells, before finally hitting the photoreceptor cells at the very back. Once the light reaches that final layer, it interacts with two highly specialized classes of photoreceptor cells, rods and cones. Rod cells are engineered for scotopic, or twilight vision. They achieve this using a specific purplish-red protein called rhodopsin, which is highly sensitive to low-light conditions. Cone cells handle photopic, or daylight vision. They are strictly responsible for color detection and sharp visual detail. To process color, the human eye uses 3 specific types of cones, each containing a different photopigment. Erythropsin responds to red light, chloropsin to green light, and cyanopsin to blue light. When incoming light stimulates all 3 of these cone types simultaneously and equally, the resulting neural combination produces the sensation of white light. Both the rhodopsin in the rods and the iodopsin pigments in the cones share a common chemical origin. They are biological derivatives of vitamin A. The entire retina functions as a high-density sensory mosaic. It takes the sharply focused light from the lens and filters it into millions of discrete data points based on intensity and spectral wavelength. The actual mechanism of vision begins with a microscopic chemical event. Light energy physically strikes the photopigments housed within those rods and cones. These photosensitive compounds contain opsin, a protein, and retinol. When light hits, it induces a dissociation. The retinol molecule physically splits away. This unzipping immediately alters the cell, changing the membrane's permeability and opening ion channels. This rapid shift in membrane permeability generates a local potential difference, a tiny electrical charge within the photoreceptor itself. Because of the retina's inside-out design, the resulting electrical signal must now reverse course. It travels outward, leaving the photoreceptor and moving forward into the layer of bipolar cells. The bipolar cells receive the charge and relay it 1 layer further forward, transmitting the signal into the ganglion cells. It is here, within the ganglion cells, that the localized potentials combine to generate a true action potential, a full-strength electrical impulse. These action potentials are then collected, bundled together into the optic nerve, and transmitted directly to the visual cortex of the brain for final processing. Transduction is the chemical bridge. It is the exact moment where physical light is disassembled and translated into the biological currency of electricity. All of those electrical signals exit the eye through the optic nerve. This exit point, located slightly above the posterior pole of the eyeball, is also where the retinal blood vessels enter. The text in this section identifies a critical limitation. This specific point is called the blind spot. Because the optic nerve fibers monopolize the space exiting the eye, there are 0 photoreceptors present in this region, making vision impossible at that exact coordinate. Just lateral to the blind spot lies a completely different piece of retinal geography. There is a yellowish pigmented spot called the macula lutea, which contains a central pit known as the fovea. The fovea represents the absolute peak of visual acuity, the point of greatest resolution. This is because the fovea is a thinned-out portion of the retina containing only densely packed cone cells, allowing for maximum detail capture. From the avascular cornea to the chemical unzipping of rhodopsin, the anatomy operates entirely on sequence. Structural transparency delivers the photon. Molecular dissociation translates it. Vision is the emergent property of an intricate system where physical engineering and biochemistry work in perfect synchronicity.